Welcome to the hidden world inside your oven. We often think of baking as an art, but fundamentally it is a science. Specifically, the chemistry of baking is defined by the interaction between yeast and gluten. When we mix dough, we aren't just combining ingredients. We are initiating a controlled sequence of biological and chemical reactions. From the moment the water hits the flour to the final rise in the oven, we are orchestrating a complex molecular dance. To understand this process, we must first identify the key ingredients and the specific roles they play. First, we have flour. It is more than just a powder. It provides the essential starches and proteins required for gluten formation. Next is water. Water acts as the trigger. It hydrates the proteins and activates the dormant yeast cells. Speaking of yeast, this is our biological leavening agent responsible for producing gas. Finally, we add salt. While it adds flavor, chemically, it controls yeast activity and actually strengthens the gluten structure. Let's zoom in on the biological engine of bread, the yeast. It is crucial to remember that yeast is a living organism. Specifically, it is a single-celled fungus with a very specific job: converting sugars into gas and alcohol. Like any living thing, it has survival needs. To activate and do its work, yeast requires moisture, warmth, and a source of food. When we first mix our dough, the yeast begins its metabolism through aerobic respiration. This occurs because oxygen is plentiful during the initial mixing stage. The chemistry here is straightforward. Sugar plus oxygen yields carbon dioxide, water, and energy. While there is some initial gas production here, this phase primarily focuses on cell growth, allowing the yeast population to multiply before the real work begins. As the oxygen within the dough is depleted, the yeast switches gears. We enter the stage of yeast metabolism known as anaerobic fermentation. This happens when oxygen runs out during the proofing stage. Now the equation changes. Sugar is converted into carbon dioxide, ethanol, and energy. This is the critical moment for bakers, as this is the main process responsible for both leavening the bread and developing its flavor. Let's analyze the products of this fermentation process. First and foremost is carbon dioxide CO2. These gas molecules get trapped by the gluten network, creating bubbles that cause the bread to rise. Secondly, we have ethanol, or alcohol. While this contributes significantly to the aroma and flavor, most of it will evaporate during baking. Finally, the process produces organic acids, which add complexity and a distinctive tanginess to the bread. While yeast provides the gas, the flour provides the container. This brings us to the chemistry of gluten proteins. Flour contains two main storage proteins, glutenin and gliadin. It is important to note that dry flour does not contain gluten. Gluten only forms when these specific proteins hydrate in water. To understand dough, we must distinguish between glutenin and gliadin, the two components that make up gluten. Gliadin is the plasticizer. It is responsible for the extensibility and stretchiness of the dough, allowing it to expand. In contrast, glutenin provides the strength. It is responsible for elasticity and structural integrity. These two are held together by disulfide bonds, which are the chemical links connecting both protein types together. How do we turn a wet mess into a structured dough? Through gluten development, hydration, and kneading. First, hydration allows the proteins to uncoil and move freely. Then comes kneading. This physical force stretches and folds those uncoiled proteins. This mechanical action is vital for bond formation, as the force encourages strong disulfide bonds to form between the protein strands. The result of this hydration and kneading is the gluten network. What was once a chaotic mixture is now an organized, interconnected protein system forming a three-dimensional viscoelastic network. This network is crucial because it is strong and flexible enough to trap the carbon dioxide gas bubbles produced by the yeast, preventing them from escaping into the air. Now we enter the proofing stage, which is essentially the trapping of CO2, or the rise 39. It happens in 4 steps. First, the dough rests in a warm environment, 40. Second, the yeast produces CO2 gas via fermentation, 41. Third, the elastic gluten network expands, trapping the gas, 42. And finally, logically, the dough doubles in volume like a balloon. Yeast isn't the only way to make things rise. Let's contrast biological leavening with chemical leaveners, starting with baking soda. Baking soda is sodium bicarbonate, NaHCO3. It acts as a base, requiring an acid to produce CO2 immediately. The reaction is simple. Base plus acid yields salt, water, and carbon dioxide. Unlike yeast, this happens instantly. Then we have baking powder. The key difference here is that baking powder contains both the base, NaHCO3, and the acid components pre-mixed. Most commercial baking powder is double-acting. This means it releases a CO2 burst upon initial wetting and then releases gas again when heated in the oven. Once the dough enters the oven, we reach the baking stage, defined by heat and structure 52. 3 things happen in sequence. First, we get oven spring, where yeast activity spikes before dying and CO2 production accelerates, 53. Next is coagulation, where the proteins, gluten, set and harden the structure, 54. Finally, gelatinization occurs, where the starch absorbs water and swells inside the dough, finalizing the texture, 55. As the baking continues, surface chemistry takes over. We see the Maillard reaction. This reaction occurs above 140 degrees Celsius on the dough's surface. Chemically, amino acids react with reducing sugars, creating the golden color and complex flavor associated with baked goods. If the temperature rises even further, we experience caramelization. This occurs at higher temperatures, specifically around 160 degrees Celsius. Unlike the Maillard reaction, this process involves sugars breaking down without protein involvement. This creates a deeper brown color and introduces sweet, nutty flavor notes to the crust. Deep inside the loaf, a critical threshold is reached. This is called the kill step, 63, when the internal temperature reaches 57 degrees Celsius to 60 degrees Celsius. The yeast is effectively killed, 64. At this point, fermentation stops completely, and the structure is permanently set by the hardened gluten and swollen starch. But what happens if we remove wheat? This is gluten-free baking chemistry. Gluten-free flours lack the elastic protein network of wheat flour. To compensate, bakers must use substitute stabilizers. Ingredients like xanthan gum, guar gum, or psyllium husk are added to mimic gluten properties and provide the structure that glutenin and gliadin usually would. Even with good chemistry, failures happen. Troubleshooting is necessary when dough fails to rise. Several variables could be at fault. Perhaps the water was too hot and killed the yeast cells instantly. Maybe there was too much salt, which inhibits yeast activity. It could be that there was not enough sugar for yeast fuel, or commonly there was insufficient kneading, creating a weak gluten network that couldn't hold the gas. In summary, the final structure of bread is a marvel of chemistry. 75. It is essentially a stable foam created by biological carbon dioxide gas trapped in a chemical protein network. 76. The exterior beauty is thanks to the Maillard reaction and caramelization, which provide the final flavor and color. 77. From a simple mix of 4 ingredients, we create something extraordinary.